Now, remember, for folks who've been here before, the COVID-19 virus is covered by spike proteins, these red proteins here on the outside of the virus. And it uses these spike proteins like little keys to enter into our cells. Now, if we zoomed in on one of these spike proteins with a microscope, this is what we would see. Now, it looks kind of weird, doesn't it? This is what we call a space-filling model of a protein. And there are 3 different colors because the protein is actually made of 3 different pieces. And we'll talk about that later. Now, this spike protein is probably the best studied part of the virus. It's what most of the vaccines are focused on. Once someone gets vaccinated or develops natural immunity from having had COVID-19, the antibodies that they make are going to be targeted at this spike protein. So it'll be useful for us to get to know the spike protein just a little bit better. The top part of the protein is what's known as the receptor binding domain. And that's because this is the part of the protein that binds to the ACE2 receptor, hence the receptor binding domain. And those antibodies that you might be making if you've already gotten the vaccine or had COVID, well, it turns out that this is where most of those are binding to as well. And I've drawn the antibody here sort of approximately at scale. And as a metaphor, it might be helpful to think about this antibody recognizing the receptor binding domain of the spike protein a little bit like your hand grabbing a shoe. It's a similar size. And you could imagine maybe you're reaching around in the dark trying to find a specific one of your shoes, and you might grab the top of the shoe, you might grab the side of the shoe, you might grab the other side of the shoe. And so that's sort of how these antibodies are really latching on and recognizing this part of the spike protein. Now let's see what happens when COVID-19 gets inside a person who's not making antibodies. Here we have an ACE2 receptor on the surface of a cell on your lungs. Almost all of your lung cells and cells in your mucous membrane have these ACE2 receptors. They're all over your body. Now let's say you inhale some virus from someone that coughed nearby. The spike protein on the surface of that virus goes and it actually really grabs on, it latches onto that ACE2 receptor, and you can see a little bit what's called a conformational change here, where other parts of the protein change their shape a little bit because of this interaction between the ACE2 receptor and the spike protein. And after that, the cell lets that virus right in. So that's no good. Now let's see what happens if you're making antibodies. So here we've got our ACE2 receptor on a lung cell again. Now the spike protein has an antibody attached to it, like that hand grabbing onto the shoe. And this antibody will physically block that spike protein from binding to that ACE2 receptor. And this would be like maybe you have your hand on your shoe and then your dog is trying to bite your shoe or something, but you're blocking it because your hand is there. And because that antibody is there, that spike protein is not able to latch onto that ACE2 receptor, and the virus does not infect the cell. So that's great. That's what we want. Now, how are these new variants gonna change this picture? And just as a local note, the first UK variant was actually detected at UC Berkeley just this past week, and it's been seen in several people in Alameda County. So because these strains, as we'll see, are more transmissible, they might quickly become the dominant strain in Alameda County and all over California. So there are about 3 major strains that we've been hearing about from the UK, South Africa, and Brazil. And we know the most about the UK strain. We've seen it for the longest time. We know that it's about 50% more easily spread from person to person, and we'll see a little bit why that may be. And it's possibly more deadly. Some studies suggest it's 30% more deadly, although it's hard to say. But just the fact that it spreads more easily is going to result in more deaths, even if it's not deadlier once you get it. And we also know that this strain has several mutations, and we're going to talk about what that means in a second, but one of these mutations is called N501Y, and we'll talk about that more. Now, the South Africa variant or strain, we know a little bit more about, or we know a little bit about, not as much as the UK strain. It's possibly more transmissible, and what's perhaps most concerning is that it seems to be better at evading antibodies from vaccines and from people who have natural immunity from having had COVID already. So some people are getting reinfected. There's a lot of concerns around that. And it also, like the UK strain, has several mutations, and that includes that N501Y mutation, just like that UK strain, but also a mutation called E484K. And we'll talk about that in a second as well. And then finally, we have the strain first identified in Brazil, and we don't know much about it, but it does share 2 mutations with the South Africa strain. And so we sort of assume it may be similar. Now, just as a side note, in America, we tend to name things that we don't like after other countries. But I'm sure we also have our own weird strains in the US as well. Unfortunately, we are doing very, very little sequencing, so we're kind of flying blind. About a month ago, there was a lot of talk about a California strain. That seems to have died down because we're just not doing a lot of sequencing, so we don't know. But I just wanted to mention that it's not like these other countries are somehow making strains and we aren't. I'm sure we're making strains too. Okay, so let's talk a little bit about what these alphanumeric codes really mean. What do these mutations mean? So let's remember how that spike protein gets made that we talked about earlier. There's a gene in the coronavirus genome, the COVID-19 genome, and that contains the information to make the spike protein in the form of RNA. This RNA gets translated into amino acids. And these are small molecules that are attached in a long strand. And these amino acids are really the building blocks of a protein. This strand then gets attracted to itself through weak chemical forces. And that results in this blob-like structure that we call a protein. And so that's what happens for a normal spike protein. I'm not showing a spike protein here on the right. This is some other protein, just as an example. Now, let's say there is a mutation in that virus genome, and that's what we've been seeing with these new strains. And you know, genomes, the virus genome, when it's infecting new people, is constantly replicating, just like the cells in our body. And so mistakes get made. So just in the way that you might have a mutation in one of your cells that leads to a higher risk of cancer, the virus genome is also mutating. This mutation then in the virus genome, and there are lots of them, lots and lots that have been recorded. But one of these mutations may result in a different amino acid being made. And so this strand of amino acids, one or maybe more of those amino acids are going to be a little bit different. And that means when that protein eventually folds, it's going to fold a little bit differently. That blob-like structure is going to be slightly different. And so specifically, we've seen this N501Y mutation, and this might look like a sort of cryptic code at first, but it's really not. In fact, it's very helpful for researchers to understand what's going on. This literally means that there was a mutation at the 501st amino acid in this long strand, which was originally an asparagine, which is what N is the symbol for. That's a common amino acid, and that amino acid is now in the mutant strain a tyrosine, which is what Y is the symbol for. So that's sort of what these alphanumeric codes mean. And so let's sort of put this all together and see what these mutations that I mentioned before look like on the spike protein. So here I'm showing what's often called a ribbon diagram of part of the spike protein. So I apologize, it's a little different from the image that I showed before, but hopefully you can recognize that there's this ACE2 receptor here in green. And here we're seeing the UK strain and the location of that N501Y mutation. And we're also seeing a deletion, but let's not worry about that right now. That N501Y mutation, you'll notice that we're seeing 3 of them, and that's because, as I said earlier, the spike protein is actually made of 3 identical amino acid strands, three proteins that come together, and so that's why there's 3 of them. But you can see that one of these mutations is right next to that ACE2 receptor where that binds, and recall that's what's so critical, that when the spike protein binds to that ACE2 receptor, that is its gateway into the cell. And what people believe is that this mutation, this N501Y mutation, right next to that ACE2 receptor, that is making it so that the spike protein sticks just a little better to that ACE2 receptor. And that's what's making it spread more easily from person to person. And maybe the idea is that you don't need as much virus. You don't need to have inhaled as much virus or been exposed to as much virus to get sick as you did before, because this new strain is a little stickier to your ACE2 receptors. And that makes it more easily spread from person to person. Now, on the right, you see these 2 mutations that are important in the South Africa and Brazil strains. And they contain also that N501Y mutation. So that means it is possible that it will spread more rapidly, although in practice, we have not really been able to evaluate that. But they also contain this really nasty E484K mutation. And that you can see is also right there next to the ACE2 receptor. In this plot, it almost looks like it's in the ACE2 receptor, but it is part of the spike protein in gray here. And you can see that, and so remember that not only is this receptor binding domain where the ACE2 receptor binds, but it's also where most of those antibodies bind that your body is making. And researchers believe that this E484K mutation may contribute to the fact that those South Africa strains are recognized less by antibodies. And thinking back to the shoe example, if you were feeling around, if your hand is an antibody and you were feeling around in the dark for a certain shoe, you might be able to find that shoe, but if I started changing your shoes a little bit, if I started modifying them, you might not recognize your shoe anymore. If I cut the tongue out of your shoe, or if I took the shoelaces out of your shoe, if I started changing that shoe, you might feel, and you might think, this isn't my shoe, this is something else. And so you're not gonna grab onto that shoe. And that's exactly what's happening with this antibody. It is not recognizing that receptor binding domain of the spike protein anymore. So that part of the protein that the antibodies used to know really well has changed. Okay, so what does this mean for the pandemic response? What does this mean for us? Well, many of the strains, as I said, seem to be more contagious. And that means that we're gonna need to take more precautions than we did before. And I'll talk about this later in the seminar about how we can maybe wear masks a little better. But what I think is more concerning to people is that there seems to be evidence that the vaccines are less effective for the South Africa and Brazil strains. And despite all the hype on all the attention this is getting, there actually isn't a ton of data on this. But unfortunately, what we do have, it all points in the same direction. So we have pretty limited data from Moderna and Pfizer. Moderna did one study here where they took antibodies from individuals who had been vaccinated with the Moderna vaccine. And they tested these antibodies against the new strains. And here on the left, these red circles are the sort of represent the original COVID strain and how well those antibodies are sticking to that strain. And on the right, these white triangles represent how well the antibodies stick to the UK strain. And so you can see there's not a big difference. Now, let me show you for the South Africa strain. and I didn't get into the codes for them all, but they all have these code. And South Africa is B1351, whereas the UK is B117. And you can see for South Africa, it's the antibodies are performing significantly worse. They're not sticking as well to that modified spike protein. And now I would expect that this would be similar for the Pfizer vaccine. Now, this is a drop, but it's not terrible. It's not down to 1 here. So there will be some degradation of performance for the vaccines. It's not possible to translate this information, though, directly into how effective the Pfizer or Moderna vaccine will be against the South Africa strain or the Brazil strain. So we still don't know, and those studies are happening, and it'll take some time until we know that information. It still may be quite effective. I wouldn't be surprised if it was still very effective. And that's what we're seeing from some of the newer vaccines, which were actually tested against the new strains because their phase 3 trials were more recent. Now they showed a similar trend where efficacy in the US and UK, so this is the Johnson & Johnson in light blue, the Novavax vaccine in dark blue. Results from both of those have come out, although they're not fully published, so we're still waiting a little bit. But the performance in the UK and US is around 75%, which is really still very good. It's not the Pfizer and Moderna quality, but it's really, really good. And then you can see against individuals in South Africa, where they assume many of those individuals have the South Africa strain, the efficacy, it drops to around 50%. But that's still really good. I mean, if all of a sudden 50% of people were immune to COVID-19, we'd be in good shape. I mean, I want to say that is still really good. Now, because of that, some of the vaccine developers are talking about developing versions that are targeted at specific strains. But this will take time, and we can talk about that when and if that happens, if that really becomes necessary. But I do want to emphasize that although a lot of the focus has been on these antibodies, and that's what I've talked about today, that's not all that we've got. As we saw in that first lecture, we have 2 types of memory T cells. We have memory B cells that are capable of making more antibodies and different antibodies. So there are really a lot of players in the immune system. And even if these antibodies that we have now are just a little bit effective, they can buy our bodies more time. So I would say, and I'm not a professional, but I have listened to some talks by professionals, and some people are more concerned than others, but there are several researchers who really know this stuff very well and are still pretty optimistic. They think this is a setback, but a small setback. And so that's what I want to communicate today. That even with these new strains, we're, I would say, only in slightly worse shape than we were before. And that is the end of my presentation for now. I'm going to pass it off to Crystal.